All right, good afternoon, everyone. Welcome back. So we are entering here the last session, I guess, of the show with 4 more talks. And so first in this session is Caroline Gauchotte-Lindsay. So the floor is yours.
Thank you very much. Well, thank you very much. Thank you very much for having me. Those of you who have attended the online version of these events know there's nobody happier than me than there is an in-person meeting. So I really hope you can all hear me properly and I don't have to revert to ancient solutions. Anyway, what I come to talk to you about today is the work we do in our research group. We're a little bit different than many of you. We're based in an engineering department, so we work very closely with engineers, mostly in water and environment. And so want to start with the acknowledgement with all the former and present lab members, and then the ones that you see in dark are students and postdocs that have been involved with the project I'm presenting just now. And then I'd like to thank some of our collaborators on the project as well. So the things we're trying to work towards is going beyond monitoring with advanced analytical tools and try to think how we can use advanced analytical tools to help with optimization and prediction and prediction of failures in engineered systems. So what do I call engineered systems? We've worked in a variety. We do a lot of work in biotechnology, so drinking water treatment, wastewater treatment, but also bioremediation of contaminated land. But we've also worked, and some of the results I'm going to show you, we've worked on a process called underground coal gasification, which I would like to reassure everyone isn't something that's going anywhere because it's extremely polluting, but we had to work on it to figure this out. And so what we are trying to do is, as you can imagine, we're working with complex samples, and then they go through a process, and through that process, they are altered into different, their signatures are changed, and can we use those signatures as a way to tell us about the health of a system, and then eventually, and also potentially identifying, and I've heard this a few times already from other speakers this last 3 days, but identifying markers of that health so that we can, you know, as a monitoring device, we can go down to more simple techniques. So the idea is we look at the whole system, we describe it first, and see if we can use it for prediction, and eventually, because we'll have to get funding and we have to use buzzwords, we are looking at digital twins. So how can we have a digital version of a system that we can inform with testing of the samples, and can this help us predict when an intervention needs to happen on a system, for example? So all this started quite a while ago when I was doing my postdoc, and this is the sort of, some of it is the results of Laura McGregor's PhD. And we were doing something different. We were doing environmental forensics. We were looking at coal tar that had been left on historical site of manufactured gasworks. So soil that was contaminated with coal tar and like pure coal tar samples. And the idea was to see if we could use a GCxGC signature of those coal tar samples to sort of date when the contamination had happened, because that's how we could find the person that would be responsible for remediation. And what we found when we did this is that by selecting a few compounds, I think it was about 242 at the time, we were going manually looking at compounds, and we found that by doing unsupervised PCA, that the way the samples were from different sites were clustering with each other was based on the manufacturing process from the tar, which is, we were working on tasks for which we had that information. So the thing that you see, CO, HR, LTHR, VR, those are like shorthands for the different processes. If we see up here, can we put it? I'm not going to try anything too complicated. This is the transformation process, like how the coal was used to make gas, and then in the process tar was formed, and depending on how the retorts were treated, the temperature, the geometry, then we would have different signature for the tar. And so looking at, and this is on samples that had been in the environment for 50 years. So even heavily wet samples still carried a fingerprint of the process that had formed them, which also means that those samples were coming from different sites. So the coal that would have been used for the formation of the tar would have been different. So that not only the coal, original coal, was influencing the signature, but also the process. And so this is where we started that thinking of, can we use the signature of the altered of a sample that's gone through a modification to relate it to the processes, the engineering processing. So we've worked on a big European project on underground coal gasification. So underground coal gasification is when you actually ignite the coal directly in the seam to produce syngas, but you have to inject water for this. You have to inject heat and you have to inject water. You have to inject oxygen and other gases. And therefore, with the gases that comes out, with the syngas, you also get processed waters. And those processed waters will contain some coal tar. They will also be polluted themselves with a lot of pHs and compounds that are associated with burning tar. And so we worked on with the mining ministry in Poland on an in situ experiment. So we had an ex situ experiment. We had a massive tank where we were doing a controlled gasification. So that wasn't in an actual seam. It was in a reactor that was modeling the seam. And at the end of this, gas was coming out, but also processed water. And so we had all this information on the process, because that's what the people were studying there. And then we were given the processed water and the tar to look at. Actually, there was virtually no tar products, and so we had to work with the processed water. And then we developed an analytical method to look at the processed water and whether we could eventually relate it to the process. So, but this is a very expensive experiment to run. It runs for about a week, and you can't really have a replicate because it's about, I think it was 100,000 euros every time we were running the sample. So the project, we were running an experiment. So the project was to, I think there was 8 that were run in total, but none of them were in the exact same condition. So that's a big, a big iota what we're doing, but that's also part of what we have to deal with when we're looking at real engineering application. So they were collecting samples for us, kind of composite sample every 24 hours. We developed, and I can talk to you about it another time if you're interested. We developed as comprehensive extraction as we could using high intensity visible sonication. So it's not an ultrasonic bath, it's not a probe. It's sonication that happens on the wall of the vials you're using for extraction. And then we had an extensive GC-MS analysis. Again, happy to talk about it, but today I was going to show you the GCxGC results. So we ran this on the Pegasus, and we had our 6 time points, and we could see by just looking at the bubble plots that there was things that were changing, particularly at the very end, because it was when the gas, like the process was being quenched, so things were drastically different. But if you can see, you can also see that by just comparing the polar versus the high polarity versus mid polarity compounds, we could see that there was something happening at time 3 and time 4. But that was a very qualitative way of doing this. So we have been thinking about how we could implement some chemometrics to look at this. And again, I'm going to put a big warning on this. Because we run Ioannis ran the GCxGC at the end of his PhD. He only ran it, and we don't have our own machine. We have to go to a different university. He only ran all of the sample once, so I don't have replicates. But what I know I do, what we do have is the fact that we know that our extraction was extremely replicable. And then we had, from the GC-MS analysis, we had a fairly good idea of how the different time points were clustering with each other by hierarchical clustering. So we use this to define our groups for our tiles, because we then process the sample using format of tile. So that's how we decided what sample to group together and what to leave out. Thomas said we've all heard enough about this, so I think you all know how this works back now, so I'm not going to spend too much time. So then we did this, and then because we had, so the kind of big tailing compounds that you see are phenols, we had an interesting, because we had so much phenols in the samples, we had an interesting colon combination, which had a 17 ms in the first dimension and a stabile wax, like a carbo wax in the second dimension. So we were trying to deal with the polarity of the compound. And yet, because we hadn't derivatized at this stage, we have those kind of big tailing points. And so this is how the tiles, doing a Fisher ratio threshold of 20. This is what this is like on the right hand side. You can see what all the different tiles that were selected in our feature table, and you can see the tailing quite evenly, but at this stage it doesn't matter too much. So then I noticed that by using, so we were thinking about what chemometrics to use. I noticed that when I was looking at the PCA that tiles provide, chromatograph tiles provide with, like once it's process the sample, I was really always interested in the loading plot. So I was interested, so you're doing Fisher ratio selection based on groups that you have given to the software. So you would expect that they come out in a PCA as grouped together. The groups that you have defined are the ones that are selecting for the compounds that for the tiles that you're carrying over in your table. So it's quite good that they're floating next to each other in the PCA. But what I was interested in is what sort of compounds were responsible for the separation, and also whether there was many compounds that would have the sort of kind of same profile. If you look at those bar charts, you know, you've got ones that you have compounds that were heavily present at the beginning and then none at the end, and you have compounds that are just there, the third sample, and you have compounds that are heavily present at the end. And so it's really interested about finding out those compounds that that were behaving the same as each other, to kind of understand if it could tell us anything about the process. Eventually, maybe saying which one are forming, you know, which one are disappearing, which ones are forming, and whether they're related to each other. So this is this is how a colleague from the computing science department at St. Andrews suggested we use affinity propagation clustering, not on the samples, but on the tiles, on the features. So the first thing is that we did a PQN normalization, and after the PQN normalization, we transposed our matrix, our feature table, and we're clustering the compounds, the tiles. We're clustering the tiles, not the samples. Okay, and the affinity propagation clustering is interesting because you don't have to tell it how many clusters you want to end up with. It decides how many clusters are reasonable based on constraint that you give it, and it's based on messaging between the variables. And the first thing you have to do is to kind of create a relationship matrix between your variables. And we looked at Pearson correlation because what we wanted to know is what were the compounds that were upregulated with each other and downregulated with each other, the tiles, not the compounds. And the other really good thing, I think, for what we're trying to do with affinity propagation clustering is that it also gives you, for each cluster, an exemplar. So it's kind of the tile which behavior across the different samples represent the best what is happening to all the tiles in that cluster. So you're already getting a marker just out of using this as propagation. And I'm hoping that the next slide will excite you as much as it excited me when I first managed to code it, which is we have on the left-hand side the full chromatogram of tiles, and then I then floated all the chromatogram of the different clusters. And when I saw things that were kind of coming out, so like in cluster 8, for example, things that were kind of coming out in familiar lines for GCxGC users, I thought something is happening that's working because I have compounds that are definitely related to each other being in the same cluster with each other. So things like, you know, cluster 7, cluster 8, but also the cluster 2, 3, and 4, if you kind of forget about the tailing, they also kind of family of compounds. And then the other thing I could do was using the exemplar, and I looked at the log of their normalized peak area, and you could, and I don't know if you see this really well, so that's on the right-hand side, you could see across the time points if these were going up, going down, and you see a lot of them have like a trend, and then that's completely broken with the third time point, which is something we saw on the bubble chromatogram as well. And then if you and then so you see cluster 2, cluster 3, cluster 4, which seems to be our phenols and other compounds related to each other are very close. That probably could be the ones clustered together. They have similar behavior, and then you have things like cluster 8 and cluster 7 that have very different behavior. So then you have to go inside and look at the different clusters and what they are. And so if we look at this cluster 2, it was kind of early in the chromatogram, there's a bit of tailing. And then what I also decided to code was the similarity between their mass spec, because I thought, well, if they're related to each other, if they're, say, like they're family with of homologous with increasing carbon chains, then they're going to have very similar mass spec, and hopefully I can see this. So the correlation matrix you see down there is how they all have, it's the correlation matrix between, the similarity matrix between their pairwise similarity matrix between their mass spec. And in fact, that allowed us to highlight families of compounds. And here we had the methylpyridine that seemed to be going down over time apart from time point 3. And then if we looked at cluster 7 and cluster 8, which looked like they were families just by looking at where they were situated, and they had a kind of negatively correlated behavior to cluster 2, then you could see that they were actually like quinoline and benzoquinoline. So the same, in the pyridine they were transformed by having fused rings of benzene. So probably we're coming from the transformation of pyridines into higher pHs. So this is where we're at with this. Statistically, it's a very low number of samples. We have the data for another trial, where they didn't sample at the same time interval, and they used very different gases spiked into the reactor. But there's actually this pyridine effect is already, we can see it already. So we're trying to put the two together so we have a little bit more of a statistical robustness. And also, because of the tiles selection, it's also very interesting to see all the stuff that hasn't come up in the feature table. So there's no naphthalene, there's no phenanthrene. Those seem to be quite stable throughout the process, which is also important when we try to create a big reaction. So my idea would be to be able to write a massive reaction equation for the process. That's where we're at with the underground co-gasification. And very quickly, because I'm running out of time, we're starting to do something similar on a very, very different type of sample. So this is what happens to us in our lab. We're looking at gravity filter from, eventually from drinking water treatment, but just now from wastewater. And we want to understand if we can create a depth profile of the organic matter and contaminants that are on the gravity filter. And the best thing because it's very difficult to absorb stuff that is on the GAC, because GAC is very absorbing, we thought about thermal desorption. And we had to do it like thermal desorbing 1 grain of GAC at a time. When you think of a big tank, we need to understand whether that's representative or not. And so for a few different depths and for GAC, virgin GAC, we went with 5 different grains, and this is very preliminary. But again, this is where I think Fisher ratios came into their own really, because we had high variability within classes, and so we really needed to get rid of this. And so we then went into Fisher ratio. We had to have a threshold much lower. I think we had at 10, but we still managed to get some interesting separation. Run a similar, this is the chromatogram we got, and then we run a similar workflow on the different, we had three depths, and you can see, and so it didn't make sense to represent them as a time series, but more like those box plots, because now we have several repeats, and you can see that again, we can find different compounds that behave differently at different depths. So in conclusion, we are quite far away from a digital twin yet. There's a lot of things we need to do. We need to understand the specificity of our processes. So sometimes you have access to a lot of samples, sometimes not. Sometimes you can do repeat, sometimes you can't. And so we need to think about this, what it means in terms of QA/QC, what it means in terms of importing workflow from other fields, especially metabolomics. But I think we're kind of, I think there is a real power into using GCxGC to describe engineering processes. Things that we've heard about this week about waste plastic pyrolysis oil, for example. I think not only can you help quantify what's in there, but maybe have a process that helps you optimize your system. And that's it.